Can I ask, am I audible in the back row there? How am I doing up there? Is that, okay, great. I'm putting this back up to remind you that both Lynn and Tim have various times you can meet with them to discuss material in the coursework, as well as my own office hours. Lynn has kindly offered to lead a review session next week, and she wanted to know whether Monday or Tuesday would be the more useful time. So I'm going to start by asking Monday. If that would work for you, raise your hand. Probably around 5, you think? Probably around 5. Let me ask first. Let me explain the system. If Monday would work, not necessarily it's your preference, but it would work, please raise your hand. If on wait, this is worse than a caucus. When I asked about the second one, that. In other words, the idea is you can raise your hand more than once. You can raise it for every day that would work for you. Okay? So, would Monday work for you? Would Tuesday work for you? Do you feel you have a sense, Lynn? Can we do Tuesday at 6? No, Lynn is shaking her head. 5 is apparently the time. So you would? Okay. So she's going to try for a room. She's going to try for a room on Monday at 5, and if that doesn't work out, we'll let you know what does work out. Okay? In addition, please do notice that there's times you can meet with her or Tim or me, in addition to any possible review sessions. Now, do people have any questions about the material that we talked about on Monday? Any questions so far on glycolysis? Guys, can we come back in line a little bit? Okay. I believe we ended up here on the lectures on Monday. Is that correct? Pretty much? Okay. So that was panel 34 of the chapter 10. So 34 was the last enzyme in glycolysis, pyruvate kinase, named for the backwards reaction, and it was the second of the 2 substrate-level phosphorylations. So this is where we start to actually get a yield of energy out of the reaction. The next 2 slides are more sort of overview ones that let you see the 2 halves of the pathway as more concrete things. What I've done is to borrow the drawings of these enzymes that are done as surface drawings by a very gifted artist. They're more sort of summary, so I won't walk through the 2 of them since we are a little short for time. This is the investment stage that gets you up to let me get my arrow back, pointer options. Okay, visible. That starts from the glucose investing ATP, second ATP invested, producing ultimately 3-carbon phosphorylated sugars, and then the second half of it in which this rather clever oxidation and phosphate addition sets up reactions in which we can get ATP made. So this is more of an overview than new material. One thing I'd like to draw your attention to are the free energy changes in the pathway of glycolysis. And this is now showing you the free energy changes as they are in the cell. These are not the standard free energy changes. These are the free energy changes under the conditions of the cell, the actual free energy changes. And what we notice if we look at these free energy changes, there are 3 big drops. There's one here, and one here, and one there. And large drops of free energy are usually found at irreversible steps. The first one is the hexokinase, which is the first of the 2 investment steps. The second big drop in free energy is phosphofructokinase, which invested another ATP, another high-energy phosphate. And the third of the 3 is pyruvate kinase, which actually harvests ATP. It's named in the backwards direction. Glyceraldehyde 3-phosphate dehydrogenase is not named for, does not have need a large drop because it's coupled to an oxidation step, actually. I think that enzyme name is wrong in there. Excuse me. We have a wrong enzyme in there. Yeah, that should be phosphoglycerate kinase. I'm sorry. Let's back up. Page up. I'm not quite sure why the thing will not let me change the slide, but let me just note right now, and I will file a correction. I don't know how that slipped past me. The enzyme that does not have a large drop is the other harvesting step, which should have been phosphoglycerate kinase, not GA3-PDH. And I apologize for not catching that in preparing it. So, but the underlying reaction there is still addressing what's going on in GA3-PDH because GA3-PDH couples an oxidation and an addition of an inorganic phosphate, that it's possible for phosphoglycerate kinase to do what it does. So let's go on down. We've talked a little bit about the regulation of glycolysis, but not completely. And there are a couple of other steps where glycolysis is regulated. We mentioned that phosphofructokinase is at the center of the pathway, so to speak, in terms of regulation. But the other two steps turn out also to be regulated because glycolysis has intermediates connected to so many other pathways. And one of the other key regulation steps is hexokinase. And hexokinase has a related enzyme that has an interesting relationship kinetically and in terms of regulation. So hexokinase takes glucose and makes a glucose 6-phosphate. And it's feedback regulated. So glucose 6-phosphate will itself inhibit the activity of the enzyme hexokinase. So this is a simple kind of feedback inhibition. There is another enzyme which catalyzes the same reaction, so glucose to glucose 6-phosphate, but it's called glucokinase, and it does not have feedback inhibition. Glucokinase is found in the liver. The relationship between these two enzymes is kind of interesting. Hexokinase has a greater affinity for glucose, whereas glucokinase has a higher final Vmax. And so the hexokinase is good at using glucose locally for a tissue and its own tissue demands, whereas the glucokinase is good in the liver as a way for the liver to sequester or store up excess glucose that's beyond the current needs so that the liver can either make glycogen or fatty acids out of it. Although hexokinase in tissues is regulated, it is not the principal regulation for glycolysis, even though it's at, quote, the beginning, if you like, of the pathway. And that's because the product of it, glucose 6-phosphate, can be used for other pathways. It can be used for glycogen synthesis. It can be used for the pentose phosphate pathway. And so it is not as committed a step as phosphofructokinase is. So it is regulated, but it is not the principal regulatory step for glycolysis. And this is giving you a little bit more details about the relationship between glucokinase and hexokinase. Hexokinase got its name because it can actually phosphorylate many sugars, not just glucose, whereas glucokinase is more specific. It has a lower affinity or a larger Km, but it can do more when it's fully saturated. It has a higher Vmax than hexokinase does. The third step in glycolysis that can be regulated is at pyruvate kinase, the last of the 10 enzymatic steps. And the regulation of, that didn't quite come out right. I think the projector has faked me out again. The regulation of pyruvate kinase is different depending on the tissue and the organism. If you're talking about the liver, remember that the liver has as an overall goal, as an organ, for homeostasis, to make sure that everything in the body gets what it needs. And so the liver is going to try to make sure that the brain gets glucose, that excess energy gets stored, and so forth. When blood glucose levels are high, that can stimulate phosphorylation of a pyruvate kinase, which is going to be a less this diagram is a little bit misleading here. High blood glucose is going to stimulate the dephosphorylation of the pyruvate kinase, so you have a more active enzyme, and it's going to be more able to carry through the rest of that pathway. Under low blood glucose levels, this enzyme is going to become phosphorylated at the cost of an ATP and then be less active. In the tissues, you can see metabolite control. And so these are a couple of examples of metabolite control that is seen in tissues. Fructose 1,6-bisphosphate can stimulate it. So if there's an accumulation sort of roughly at the place where we're shifting from the 6 to the 3 carbon, we can stimulate the activity of the downstream part, get that metabolite flux through. On the other hand, ATP or alanine, which are both signs of either, in the case of ATP, excess energy, alanine, abundant amino acids, because they're rich in the bloodstream at the moment, whatever, can inhibit that and divert it to other things. Glucose is not the only sugar in the world, and I'm not going to cover these in quite the detail I might on when we have 3 full lectures, but I do want to touch on them. One important one is lactose, and lactose tells you something important about it, which is it's from sugar. Lactic, lactating, all those things are related to milk, and lactose is the milk sugar. The Milky Way is also called the galaxy, so we see it there too. Now, lactose is interesting because it's a disaccharide, so it's 2 sugars joined together. It happens to be 2 hexoses joined together. And lactose is specifically glucose plus a galactose. And I don't expect you to know the structure of galactose. Lactose is not itself directly absorbable. And so in the gut, there's a lactase that can split lactose into the 2 sugars, unless, of course, you happen to not keep that enzyme into adulthood. Most Americans can tolerate milk as adults, but some can't. And in many parts of the world, most adults can't tolerate milk as an adult, and it's because of the lactose in it. In something like yogurt, the microorganisms have done the work already of breaking the lactose into 2 fragments and partially breaking down the sugars. And so something like yogurt can be tolerated by people who are lactose intolerant in many cases. Galactose is not itself glucose, but it's a free reaction to isomerize it into a glucose, and therefore it provides the same amount of energy as glucose itself. Another important sugar is sucrose. Sucrose is so common that it basically has the name the sugar. And it is the most common disaccharide in plants. And this is what it looks like. It's a disaccharide of glucose and fructose. Fructose being, loosely speaking, fruit sugar. We'll see more about that in a little bit. If you have table sugar in a sugar bowl, it's sucrose. And it is readily digested in the intestines. There's no adult deficiency like lactose. Now, what's interesting about it is that in the linkage between the 2 sugars, it sort of covers up the reducing group that's visible on many other sugars. And so sucrose will not chemically attack other things. Glucose can become covalently attached to proteins without the presence of any enzymes. It's one of the problems that diabetics have, is that the glucose at high levels can attach itself to body proteins. Sucrose, by contrast, cannot undergo that kind of reaction, and that may be why plants use it extensively for both transportation and for storage. Once we get it into our body, it'll get broken down into glucose and fructose. Fructose. So fructose is fruit sugar, loosely speaking. We get some of our fructose from sucrose, where it's half the molecule. It's also a common sugar in fruits. Gram for gram, Fructose is sweeter than glucose, and it's also more soluble than sucrose. And so those are advantages compared to those sugars in terms of the economics of it. And we now have fructose available in quantities that were never before seen through people like Archer Daniels Midland, who can produce high fructose corn syrup. They start with starch, which is all glucose, and use enzymes to turn some of the glucose into fructose. It's not clear that fructose is a great thing in large quantities. Small quantities probably fine. If you think about hunters and gatherers, the number of times they could eat very much fruit were not very many. Fructose was an exciting, nice part of their diet, but not a big part of their diet. In our diet, it can be a significant fraction of the calories if you like drinking soda and stuff. And part of the reason it may not be quite as good a thing is that it bypasses some of the regulation that controls how glucose is used in the body. And let's take a little look at how that would happen. Here we start off with a fructose molecule. It's a 5-membered ring here. There's a special enzyme called fructokinase. And fructokinase, again, adds a phosphate, as you might expect. So now we have fructose 1-phosphate. Fructose 1-phosphate is the substrate for a separate enzyme called fructose 1-phosphate aldolase, which can split it now into 2 fragments, rather like aldolase did for 1,6-bisphosphate fructose bisphosphate. And now we have a dihydroxyacetone phosphate, which we would have normally, and we have a glyceraldehyde, no phosphate on it yet. So there's a separate enzyme, triose kinase, that can take that glyceraldehyde and add a phosphate to it. So we're now at the point where we have 2 3-carbon sugars, but we've come in downstream of hexokinase. We've come in downstream of phosphofructokinase. We have bypassed 2 of the 3 regulated steps of glycolysis. And so it's kind of slipping in by the back door. We see that especially in the liver, where the fructokinase is more abundant. Muscles and fat tissue have more hexokinase than fructokinase, and so it probably isn't as bad. But the liver is, the organ that is in charge of things like making a lot of the fatty acids and balancing stuff, so it has potential for bypassing regulation there. Fructose itself is principally metabolized by the liver. It can be phosphorylated by hexokinase, but it's not carried into the cells as well as glucose, and it's not as good a substrate. So the majority of the fructose will, in fact, be metabolized by the liver. And that's the place where I said it's going to be sort of slipping in below the controlled enzymes. One consequence of that is when you look at the carbons of that, they may well end up being preferentially used for fat synthesis rather than for contemporary energy demands. I've had friends who are diabetics who were asked to use an experimental high fructose diet who found they had much less energy on it than they had when they were going about it through a more conventional balance of sources. Well, we got through pyruvate, and at the end of, we got through glycolysis, and at the end of the 10 steps of glycolysis, we had a 3-carbon fragment. We had pyruvate. And this is just to refresh your memories of what pyruvate looks like. Methyl group at one end, carboxylic acid at the other, carbonyl carbon here, very oxidized, very reduced. We're going to have to do something with the pyruvate. And what we're going to do with the pyruvate will depend on the conditions of the cell and who's doing it. Now, this is one of the things that can happen to pyruvate. It can be used catabolically. And it can be used catabolically in the absence of oxygen if we look at a reaction like this. Here's your pyruvate. Here's your NADH. Lactate dehydrogenase, which is named for the backwards reaction, can convert that pyruvate into lactate. So you can see this carbon has been reduced. And now we have an NAD plus back, and that NAD plus can go back up to the glycolytic pathway and be used again and again for the second half of that pathway. So that this process, generating lactate from pyruvate, allows glycolysis to continue in the absence of oxygen. Another thing we can do with pyruvate is anabolic. And pyruvate is a precursor for an awful lot of things. We can make fatty acids out of it. We can make amino acids out of it. We can feed it into the citric acid cycle. The citric acid cycle itself can be anabolic or catabolic, but we'll get to that point again in a little bit. We've mentioned it both in that overview of metabolism, but we'll get back to that. So here we have pyruvate. If we want to feed it either into the citric acid cycle, or if we want to make fatty acids out of it, we can react the pyruvate and make acetyl-CoA. And we'll see more details of that when we get to the citric acid cycle proper. So here you can see the 2-carbon fragment here, that reaction driven by a decarboxylation. We're going to see a number of reactions driven by a decarboxylation. If you can make gas come off and you breathe it out, that can pull a reaction forward. Decarboxylations are one very powerful way to make a reaction go. Another thing we can do to pyruvate is to add a carbon dioxide. And I just told you that that will pull reactions forward. So to make that carbon dioxide go on, we have to invest energy. So if we want to make pyruvate into a 4-carbon fragment, like oxaloacetate, we can take pyruvate and carbon dioxide and ATP. And now we have pyruvate carboxylase. So this is the enzyme that adds a CO2 to it, and the product of that then is oxaloacetate. And we're going to see that oxaloacetate is itself a precursor for the citric acid cycle or for amino acids. I'll just touch lightly on this. This is the active group of the pyruvate carboxylase. Biotin is one of the B vitamins, and again, a B vitamin is playing an important role at the active site of an enzyme. I think given that we have the snow day, I'm going to skip these things on the enzyme mechanisms and go to figure 51. If we look at other ways in which pyruvate can be handled in different tissues or different organisms, we see a variety of strategies. If we, for instance, look at yeast, we see something that looks like this. You can take pyruvate, that CO2 group there can come off with an enzyme called pyruvate carboxylase. We now have a new aldehyde. There's an acetaldehyde, and that then provides a substrate for alcohol dehydrogenase to regenerate NAD+. Previous time I showed you how making lactate would regenerate NAD+. In yeast, we do it by making ethanol. So clearly, this enzyme is named for the backwards reaction. But ethanol is just a byproduct in yeast of anaerobic metabolism. They happen to be among the few organisms that can tolerate living in several percent alcohol. Humans die at 1% or so, whereas yeast, almost uniquely among organisms, can live in alcohol up to over 10% of the volume. So this is a good strategy for them. In us, that ethanol would have to have something done to it, and the pathway in which we would metabolize ethanol would involve using alcohol dehydrogenase, and the name it's directed to acetaldehyde, and then a second dehydrogenase reaction to acetate. But you'll notice that both of these are reactions that produce NADH rather than NAD+. And so high levels of ethanol can act as a sink for the NAD+ that's needed for glycolysis. So if you have high concentrations of ethanol, the acetaldehyde cannot get metabolized completely, and that's one of the major places in which ethanol makes nasty things happen when it's excessive. All right. New pathway. I want to talk now about a pathway that is sort of a Siamese twin, if you like, of glycolysis, or dark side, light side kind of relationship. Luke, I'm your father. But in this case, it's a synthetic pathway. So gluconeogenesis is going to be an anabolic pathway. Its name tells you what it's going to do. Gluconeogenesis. Neo, making something new. Genesis, making something new. Glucose, sugar. So gluconeogenesis makes new glucose. It's going to make new sugar. And it's going to make the sugar from two things. It's going to use the backbones, the carbon backbones from amino acids, and it's going to use energy from fatty acids. And you could use energy from the amino acids, but there are very few times that you have so many amino acids to spare that you use them for energy. So the more common thing is to use amino acid backbones and fatty acid for lots of energy. And why would the body do this? Well, it's to supply the brain and red blood cells. Now, the brain seems to have a strong preference for glucose. It's not an absolute preference, but it's a very strong preference. It takes major metabolic adaptation for it to get away from that. The red blood cells, you may remember, have hemoglobin in them. And the hemoglobin in them uses 2,3-BPG to regulate oxygen affinity. And so red blood cells have to have glucose in order to make the 2,3-BPG. So that's why those two tissues of the whole body are especially sensitive to the presence of glucose. The pathway of gluconeogenesis is most common in the liver. The liver is your metabolic organ. If you can't remember where a pathway is and you say the liver, the odds will be with you. It's the one that tries to make things balance. The kidney has some capacity for gluconeogenesis, and the pancreas has a bit, because the pancreas's job is to regulate glucose levels in the blood. Now, if you've learned your glycolysis, which I sincerely hope you have already learned almost all of gluconeogenesis, because gluconeogenesis and glycolysis share a lot of the enzymes. They're exactly the same enzymes in the same place. However, you remember that there are 3 irreversible steps in glycolysis. If they're irreversible, gluconeogenesis cannot share them. And therefore, they have to have what are called bypass reactions in order to bypass the irreversible steps of glycolysis with separate enzymes that are specific to gluconeogenesis. Now, the first step that has to be reversed, which would be phospholipyruvate to pyruvate, is strongly favored enough that it actually takes 2 enzymes to backtrack. And it's kind of an odd strategy that the body uses for that. You have pyruvate, and you have pyruvate carboxylase. Its name is a pretty straightforward name. It's going to add a carbon dioxide to a pyruvate. And now we have a 4-carbon a 4-carbon molecule, oxaloacetate. We'll see oxaloacetate again. Cost us a high-energy bond. This molecule is now the substrate for the next enzyme in this bypass step, which is, excuse me for this long name, phospholipyruvate carboxykinase. It's named backwards just to make life that much more interesting. So if you were to read it in this direction, it would be taking phospholipyruvate and adding a carbon dioxide to it. In gluconeogenesis, it's actually letting go of this carbon dioxide and spending a GTP in order to make a phospholipyruvate. Now, phospholipyruvate carboxykinase is a long name. It sometimes is called PEP-CK, PEP for PEP, CK for carboxykinase. PEP-CK is actually a very important enzyme because it's part of the regulation of carbohydrate metabolism. Since this is an essential step to gluconeogenesis, the control of PEP-CK is an important place where the body controls gluconeogenesis. And understanding the pathology of diabetes and so forth has proven to make PEP-CK a very significant target. So we bypassed one of the 3 irreversible steps. And here we're going to be looking at gluconeogenesis. Glycolysis would be coming from the top of the page down. Gluconeogenesis was going from the bottom of the page up. And you can see that this first bypass step, by taking 2 enzymes, has gotten us up back to phosphenolpyruvate. The reactions going up towards the top of the page for gluconeogenesis are perfectly straightforward. Enolase will give us 2-phosphoglycerate. Phosphoglycerate mutase will give us 3-phosphoglycerate. Phosphoglycerate kinase will cost us an ATP to make 1,3-bisphosphoglycerate. So gluconeogenesis has cost us a high-energy bond here. It's cost us a high-energy bond there, and cost us another high-energy bond there. All right? But we're working uphill. We should expect to spend energy to go uphill. Any questions so far? Okay. So here's the rest of it. We have here 1,3-bisphosphoglycerate. In this case, the glyceraldehyde 3-phosphate dehydrogenase is splitting off an inorganic phosphate, and it's producing NAD+. So we have simple glyceraldehyde 3-phosphate at the end of it. We have triphosphate isomerase that can give us a pool that has both of them in it now. Aldolase can smush them back together, and now we've got a fructose 1,6-bisphosphate. So clearly we had to start off with 2 pyruvates to do this, right? Got to make the carbons add up. Now we have fructose 1,6-bisphosphate. It's easy to throw energy away, so the second bypass step is simply a bisphosphatase. It takes one of those 2 phosphates and throws it away. So that's a very doable reaction, and it's going to be favored in one direction. Now we have fructose 6-phosphate. Phosphoglucoisomerase happily switches that backwards and forwards to glucose 6-phosphate. Again, throwing energy away is easy. The second phosphatase cuts off the other PI, and now we have glucose. So there are 3 bypass steps. It cost us several high-energy bonds to get to there. So clearly, making glucose from pyruvate is not going to be a net way to get energy. It's a way to make glucose specifically for the brain and the red blood cells when glucose per se is not available otherwise. Other questions? Okay. This is just a reminder that our second hour exam will be in a week. It'll be here, 3:30. It's possible you may need to arrive late. I understand that. We will ask people arriving late to wait, and we'll seat them at 3:40. And it will cover the 5 chapters, chapters 6 through 10. We will not explicitly cover chapter 11. We'll be starting to talk about the citric acid cycle on Friday. However, the overview of metabolism definitely had material about the citric acid cycle. Some of the stuff from chapter 10 connects to the citric acid cycle, and all of that material may well appear on the exam as well. So gluconeogenesis is an expensive pathway. It costs you roughly 4 more high-energy bonds to make a glucose than you can get out of a glucose. And so you would not want to do it unless you need to do it. You wouldn't want both of them to run at the same time, or you would end up wasting energy. Here's an example of what's meant by a futile cycle. Let's just start for the moment at fructose 6-phosphate. If both glycolysis and gluconeogenesis are running at full tilt, this is what you would get. Your fructose 6-phosphate would go to phosphofructokinase, which would make the fructose 1,6. It would then be a substrate for the phosphatase in gluconeogenesis, which would make more fructose 6-phosphate, and you could go around and around in that thing and waste an awful lot of energy if you had no way to control it. And in fact, we do have a way to control it. We've mentioned before that fructose 2,6-bisphosphate was an allosteric regulator of phosphofructokinase for glycolysis, and that it stimulates glycolysis by stimulating phosphofructokinase. It also has a second role in which it inhibits the bisphosphatase. So at the same time that the fructose 2,6-bisphosphate is stimulating glycolysis, it is inhibiting gluconeogenesis. Obviously not the same molecule. You have to have different molecules and different enzymes. But by controlling just the levels of fructose 2,6-bisphosphate, the cell controls both pathways in a way that keeps them from running futilely, wasting energy all the time. And it turns out to be a very efficient way to control a pathway because you can control it at the level of flux. And this is just an example with made-up numbers for the moment. Here we have a pair of pathways that are going in opposite directions with the potential to be a futile cycle. And at this point, if the enzyme activities are at this level, the net amount of B that gets through is 10 units. You've got 100 units going forward and 90 going back. But suppose you have something that can change the activity of both of those by not very much. So now you've got 120 units forward and 70 back. The net flux is now 48 instead of 10. You've had nearly a 5-fold difference in the flux for only, a 20% difference in each enzyme. So it gives us potential to control that way. Whenever you have a pair of reactions that are going in opposite directions, you have that potential for futile cycles. And there is some of that happening in cells. You don't want it to go full tilt all the time, or you're going to just waste energy. In a controlled form like this, it can allow you to amplify the signal from the regulator. As I said here, you can have a 5-fold increase in net flux for only a small change in the enzymes. And it can even be something that is useful per se. So, for example, there are futile cycles that allow the bumblebee to heat up its body. And a bumblebee can go out on a colder day than a honeybee can because the bumblebee uses futile cycles to generate waste heat. Only it's not waste heat, it's giving it a chance to get out and get ahead of the competition. All right. So of the pathways we've talked about so far, I've talked about glycolysis to break down glucose. We've talked about gluconeogenesis to make glucose. Suppose we have so much glucose we want to store it. Well, then we can synthesize glycogen. We can't just reverse phosphorylasis. It won't go that way. That's an essentially irreversible reaction. Glycogen breakdown through phosphorylasis is a one-way trip. So we have to get there some other way. Just like in the glycolysis, gluconeogenesis, there had to be, in effect, a bypass or an opposite, different reaction for the irreversible steps. So what happens? Let's say we're going to start with the glucose 6-phosphate that we have on hand from hexokinase. There's a lot of blood sugar, the liver's taken it up. Now I've got glucose 6-phosphate. Glucose 6-phosphate can make glucose 1-phosphate out of it. And then that glucose 1-phosphate can be the substrate for an enzyme that will add onto it a UDP tail. So you have a glucose here, and after that reaction you have what's called UDP glucose. So you can think of it as a glucose with this tail on it. And that's a strategy we're going to see a number of times. We mentioned that in the review of metabolism, that you can have activated intermediates. And this is an example of an activated intermediate. The UDP glucose can undergo reactions that glucose alone could not undergo. It does cost us something. Since this is a phosphorylated sugar to begin with, and we started with the UTP, to make the UDP glucose, we spit off a pyrophosphate, which is a double inorganic phosphate. The bond in the middle of pyrophosphate is a high-energy bond. And if it's split through phosphatase to 2 inorganic phosphates, that helps to drive the reaction. Splitting the inorganic phosphate, the pyrophosphate into inorganic phosphate is part of what provides the driving force for this reaction. We're also going to see that strategy again in many places. So UDP glucose makes a good carrier for a glucose in an activated form. And we have a glycogen, some amount of glycogen to start with. UDP glucose can then transfer that glucose onto the glycogen chain, and what's released at the end is a UDP. So if you look at that, it's going to cost one phosphoanhydride bond to add a glucose to a glycogen molecule. The glycogen synthase simply elongates this. There are other enzymes that can take part of a glycogen and move it around to create a branch, but that is a free reaction. It's freely reversible. And as you might guess, since glycogen synthase is taking up sugar to store for future reasons, it is also under hormonal control so that it's not, snatching glucose away when it's needed by the body. Okay. I think we may be a little bit brisker looking at the other sugar synthesis. Let me just see what else we have on here today, because I do think we want to get to the pentose phosphate pathway. Yeah, let's go over this to the pentose phosphate pathway. Okay. So on slide 62, we introduce another pathway. We've seen glycogen breakdown, glycogen synthesis. We've seen glycolysis and gluconeogenesis. And then we have the pentose phosphate pathway. First of all, it's a pentose phosphate, so there's going to be 5 carbon sugars in here somewhere. And secondly, it's a really multi-purpose pathway. It has several functions, not just one. It's also a very old one. I don't know that there are any organisms that are lacking it. Do you know, Lynn? The offhand? Yeah, it certainly is very old. And some of the things that it's going to do, for instance, it's going to make NADPH. You may remember that in the overview we talked about how NADH gets used for energy production, but NADPH is tagged as use me biosynthetically. Energy content about the same, but NADPH is what's needed for biosynthesis and for anabolic processes. Going to come from the pentose phosphate pathway. Pentose phosphate pathway also produces ribose. Ribose is obviously a very important component in making nucleic acids. So 2 very important anabolic functions for the pentose phosphate pathway. And the first reaction in the pentose phosphate pathway is glucose 6-phosphate dehydrogenase. So here's glucose 6-phosphate. There's going to be a dehydrogenase, so there's going to be something reduced at the end of it. But in this case, it's NADPH. And you end up with 6-phosphogluconolactone. So it's still phosphorylated. So we've seen 3 different things you can use glucose 6-phosphate for so far. Second reaction takes that closed ring and just adds water to it to open it up. And now we have 6-phosphogluconate. So it's got a carboxylic acid at the end, which is what makes it gluconate. So this is a phosphorylated sugar acid. And this end is telling you that it got oxidized a bit, right? That had been an aldehyde, and now it's an acid. So now it's been slightly oxidized. The third step in the reaction in the pathway is also an oxidation reduction step, and it also uses NADP, and it's 6-phosphogluconate dehydrogenase. You hear dehydrogenase, very likely NAD, FAD, ubiquinone. One of those electron carriers is almost certainly going to be part of the reaction. And again, in this case, it's NADPH producing a store of reduced intermediates that can be used for anabolic purposes. In this case, it's driven by a loss of carbon dioxide. You remember we talked before about how if you throw away carbon dioxide, you can help drive forward a high-energy reaction. And that's what happens here. And look at what's the product. Now we have ribulose 5-phosphate. So now we have a 5-carbon phosphorylated sugar. So now we're starting to get into pentoses. This is a pentose, finally. But so far we've made 2 NADPHs, haven't we? Ribulose 5-phosphate is energetically equivalent to ribose 5-phosphate. And so an isomerase can freely reversibly produce one from the other. Now, one of the things that you need NADPH for is for making nucleic acids. Now, you need ribose to make nucleic acids. But if you're going to make DNA, deoxyribose, you probably need to reduce your ribose to make deoxyribose. And so one of the uses of NADPH is in reducing ribose to deoxyribose. In this case, you start with an NDP. It's not just a raw ribose, but we are, in fact, going to have to take that 2-carbon and reduce it down to that. So the pentose phosphate pathway not only produces riboses for nucleic acids, it also produces the NADPH needed for the reducing. So there are a number of enzymes in the pentose phosphate pathway, and I do not expect you to know them all. I would like you to know the strategy behind the pentose pathway and also to be acquainted with these two kinds of enzymes because we're going to see them in other places. Enzymes that are called isomerase or epimerases can change conformation. So you can change ribulose into a ribose, for instance. Same number of carbons, but organized a bit differently. A second group of enzymes called transketolase or transaldolase can move carbon fragments around. And to give you sort of an idea of what you can accomplish with the freedom to move carbon fragments, this is in a very schematic sense what we're going to be trying to do through the pentose phosphate pathway. You can start off with 3 5-carbon sugars. And using transketolases and transaldolases, you can move 2 carbons onto 1 to make a 7-carbon sugar, and now you have a 3 and a 7-carbon sugar. Or you could put 3 carbons back onto that, and now you have a 6 and a 4-carbon sugar. Or you could take 2 carbons there and put it there, and end up with 2 6-carbon sugars and 1 3-carbon fragment left over. So these reactions allow you to rearrange the chain length without any expense. These enzymes are free. And so you can start with 3 pentoses and end up with 2 hexoses and a 3-carbon fragment left over. This is an overview of the things that can happen in pentose phosphate pathway. We've talked already about glucose 6-phosphate producing NADPH on the way to ribulose 5-phosphate. This region of the pathway is called the oxidative phase, and it's going to produce reducing power in the form of NADPH, and it's going to produce 5-carbon sugars. The 5-carbon sugars that can be produced at that point then undergo the non-oxidative phase and can be rearranged into a lot of other things. And if you take a look at some of these other things, some of those other things include fructose 6-phosphate. Fructose 6-phosphate sounds very familiar. That's an intermediate of glycolysis, right? And here's glyceraldehyde 3-phosphate, another intermediate of glycolysis. So the non-oxidative part of the pentose phosphate pathway allows the excess carbons to be fed into glycolysis. There are overall, one of the interesting things about the pentose phosphate pathway is you don't need to learn about which is the regulated step. There is no regulated step. The pentose phosphate activity is ebbs and flows as its products are used up. So this is one of the few cases where demand is just pulling things in and out of it as it needs to go. But the balance of what's coming out can be different. So for example, oops, sorry. There we go. For example, you can use it with rearrangements down here so that all of the glucose ends up in ribose 5-phosphate, lots of ribose. You can use it so you get a balance between reducing power as NADPH and ribose 5-phosphate. You can use it as a way to oxidize glucose completely to NADPH. So if you keep running those backbones through this loop, the carbons keep coming out as carbon dioxide, and you make lots and lots and lots of NADPH. And the fourth way of using it is in producing and reducing power as NADPH and producing pyruvate and ATP. So it's a very flexible pathway. And the next 4 slides are just showing that in larger print. So I think I will skip them for the moment and just make the point on this slide that consistent with what we're seeing there, the pentose phosphate pathway, or the pentose phosphate shunt, is very active in highly active synthetic tissues for steroids, fatty acids, in red blood cells, mammary glands. These are all tissues that have high levels of the pentose phosphate pathway to accommodate those demands. And I think the rest of what we have here, since it's 4:20 now, can be considered supplementary at this point. Yeah. Okay. There's a useful diagram for an overview. You might find this a useful review of the various carbohydrate pathways.